Hi everyone, my name is Shawin Vitsupakorn, and yeah, I'll be presenting on the in vitro synergy of Amphotericin B and Flucytosine against Talaromyces Marneffei. So Talaromyces Marneffei, or TM for short, is a dimorphic fungus that's endemic to Southeast Asia, northeastern India, and the southern parts of China. And TM is dimorphic, which means that it has two distinct morphological forms. At 25 degrees Celsius, out in the environment, it grows first as a mold. And once we get to 37 degrees Celsius, we see here TM grows as a yeast. And the yeast phase is the pathogenic form of TM in mammalian hosts. So TM causes talaromycosis, which is a leading HIV-associated opportunistic infection and cause of AIDS-related death. And despite the availability and advancement of antifungal therapy, mortality of talaromycosis is still up to 30%. So currently, Amphotericin B, or Ampho-B, is the standard treatment for talaromycosis. And how Ampho-B works is it binds to ergosterol, which is a key component of the fungal cell membrane. And so once it binds, it opens up ion channels and causes leakage of intracellular ions and eventually cell death. And while Ampho-B is very potent as a drug, it is also highly toxic. And so we really see combination therapy as an opportunity for us to lower the dosage of Ampho-B that we use for patients and in turn reduce mortality. So partial synergy between Ampho-B and another antifungal drug, Flucytosine, or 5-FC, has been established in Cryptococcus and Candida as well. And Flucytosine works by inhibiting RNA and DNA synthesis within the nucleus. And so the pharmacodynamic basis for this synergy between Ampho-B and 5-FC has been summed up in what is known as the permeability hypothesis. And basically this means that Ampho-B first pokes holes in the cell membrane, thus allowing 5-FC easier, more direct access to the nucleus, where it kind of does its magic. And this in vitro evidence of synergy between these two drugs went on to inform clinical trials that defined 5-FC and Ampho-B combination therapy as the gold standard treatment for cryptococcal meningitis. And so our study, we hypothesized that we would see the same synergistic interaction between Ampho-B and 5-FC, but against our organism of interest, TM. And in order to investigate this, we went on to evaluate the in vitro interaction between Ampho-B and 5-FC across 27 TM isolates from patients enrolled in the itraconazole versus Amphotericin B for talaromycosis, or IVAP trial. And this was conducted in Vietnam. And the two main methods, which I'll go through in the following slides, were a novel colorimetric antifungal susceptibility testing method and the checkerboard assay. So the main question that antifungal susceptibility testing answers is essentially how susceptible is an organism, in our case TM, to a specific drug in vitro. And the primary endpoint for susceptibility testing is the minimum inhibitory concentration, or MIC. And the MIC is the lowest concentration of a drug that will inhibit fungal growth to a certain degree. And so in our case, we took this to be 95% growth. Now, unfortunately, there is no standardized method for susceptibility testing for the dimorphic fungi. And so our lab has developed in-house a TM-specific colorimetric method that's been based off of CLSI guidelines, as well as a lot of optimization experiments we've performed. And so I'll walk through the key steps. First, we grew the TM yeast at 37 degrees Celsius for 3 days. Then we suspended these yeast cells in buffer and then standardized the inoculum to an optical density of 0.5 to 0.6. We added this inoculum to plates that were prepared with serial dilutions of 5-FC and Ampho-B, and then incubated these plates at 37 degrees Celsius. After 24 hours of incubation, we added Alamar Blue, and this is a dye that turns from the color blue to pink in response to cellular metabolism. And finally, after an additional 48 hours in the incubator, we took the plates out and then read the fluorescence intensity and then calculated the percent inhibition of fungal growth. To assess the drug-drug interactions, we used what is known as the checkerboard assay. And as you can see on the screen, it's an 8x8 layout with amphotericin B added in rows and flucytosine added by columns. And so noted here as PC in pink is the positive control. And so since there was no drug in this well, it allowed for uninhibited growth. And then labeled NC, the negative control, it had no inoculum, so it was our baseline reading. And I wanted to point out two main wells here, noted partial synergy and full synergy. And partial synergy is when the MIC of the two drugs combined is at least one concentration or twofold lower than the MIC of the drugs alone. And full synergy is when, the MIC of the drugs combined is at least two concentrations or fourfold lower than the MIC of the drugs alone. And so we calculated what is known as the fractional inhibitory concentration, or FIC, to assign what type of interaction was taking place between these drugs. And so the FIC is essentially this fraction of the MIC of the drug in combination over the MIC of the drug alone. And we would calculate this separately for AmphoB and 5FC, then add them together to get the FIC index, or the FICI. And different ranges of FICI correspond to different interactions here. So from antagonism, which is what we don't want, to full synergy, which is what we hope for. And we found that the geometric mean of the MICs for AmphoB and 5FC when combined were significantly lower than the drugs acting alone. Specifically, for AmphoB, it went from an MIC alone of 0.41 to 0.15 micrograms per mil when acting with 5FC. And for 5FC, this difference was from 0.21 to 0.07 micrograms per mil. And both of these differences were statistically significant according to the Mann-Whitney tests that we used. As for the interactions between AmphoB and 5FC, we observed partial synergy in the majority, 78% of the strains tested. And for the remaining 22%, we saw indifference. And it's important to note that there was no antagonism that we recorded in any of the strains. And this figure kind of just shows the distribution of these interactions. So to sum things together, this study really documents the first in vitro evidence of partial synergy that we have between AmphoB and 5FC against TM. And we really want to emphasize that combination therapy between 5FC and AmphoB has the great potential to enhance fungicidal activity while reducing patient exposure to AmphoB. And these really go hand in hand in reducing mortality and improving clinical outcomes. This in vitro study really forms the premise for clinical trials that we've designed to redefine treatment guidelines for talaromycosis and hopefully other endemic mycoses. So I want to thank the patients who donated their samples for our study, as well as the healthcare staff who helped with patient recruitment and the IVAP trial, and also my fellow teammates at Dr. Tuilei's lab. And lastly, the funders of our research, the NIH specifically for sponsoring this study, and the Wellcome Trust Department for International Development and Medical Research Council for sponsoring the IVAP trial. And thank you so much for your time.